Welcome, curious minds, to the fascinating realm of genetic mutations. I am Naraimati, and today we embark on a journey through the intricate world of DNA, where the smallest changes can have the most profound effects. Get ready to dive deep into the captivating tapestry of different types of mutations. Mutations. So what are they? Genetic mutations are changes that occur in an organism's DNA sequence. They can happen naturally, spontaneous mutations, or can be caused by external factors such as mutagens, like chemicals and radiation. Mutations can impact an organism's traits, behavior, and disease risk. They can also only be passed on if the mutation occurs in an egg or sperm cell, generally a reproductive cell. There are three different kinds of mutations: a good mutation, a bad mutation, and a neutral mutation. So let's talk about good mutations. Some mutations are good. They are beneficial and drive evolution in organisms. An example of a good mutation is from human evolution itself. Human evolution has been marked by significant changes, one of which is the transition from a long jaw to a smaller jaw, which happened as a result of course, mutations. This transformation provided more space for the expansion of the brain. This evolutionary adaptation facilitated the growth and development of the human brain, contributing to our exceptional cognitive abilities and distinguishing us from our primate relatives. Next, we have bad mutations. Some mutations are bad. If you find a mutated frog with extra limbs and extreme skin abnormalities, it is a sign of harmful pollutants and toxins in their environment and a very bad and unhealthy degraded ecosystem. And last but not least, neutral mutations. Some mutations are neutral. These are the kind of mutations we want if we were to have any, because this means that there is a mutation, but it doesn't impact us at all. Let's talk about different types of mutations. In this intricate world of point mutations, where the smallest alterations in DNA can have profound effects, there are three different types of mutations. The first one is substitution. This is when one or more base pairs are replaced by another. The second one is insertion, in which one or more bases are added to the sequence. And the last one is deletion, which is when one or more bases are lost from the sequence. Now, believe it or not, there are three different types of mutations that can occur within substitution mutations. The first one is silence, the second one is missense, and the last one is nonsense. Silent mutations occur in the introns, which are the non-coding regions of your DNA, that don't exactly code for anything, or they occur as a result of the genetic code's redundant nature. If a base is replaced by another in an intron, since that region of the DNA doesn't exactly code for anything, the mutation will have no effect as it's not important. The mutation is cut out during post-transcriptional modifications and it won't be translated. The other case is when the changed base ends up coding for the same protein as the original or correct base. The original mRNA sequence, GCC, and the mutated mRNA sequence, GCU, both code for the alanine amino acid. The more redundant nature of the genetic codes allow each amino acid to have more than one code, and since the final amino acid sequence, or in other words, the polypeptide chain, hasn't been changed, the mutation is least likely to have any long-term damage. Missense mutations occur when the replaced base pair alters the codon to code for a different amino acid. The original mRNA sequence, UGC, codes for cysteine, and the mutated mRNA sequence, GGC, codes for arginine. The effects of such a minor mutation can be deadly because polypeptide chains are primary structures that fold into tertiary and quaternary structures from the archain interactions between amino acids. When the wrong amino acid is brought in, the necessary archain interactions don't happen, and as a result, the protein is folded wrong. The structure of the protein changes, and as a result, the function also changes. This simple mutation can result in deadly diseases such as sickle cell anemia, in which the changed amino acid results in the shape of a red blood cell to look like a sickle, which gets tangled up in blood vessels, blocking them and causing serious health problems. Nonsense mutations. Nonsense mutations occur when the changed base results in a premature stop codon that results in only part of the mRNA sequence to be translated and produces completely non-functional proteins. The original mRNA sequence, UGC, codes for cysteine, and the mutated mRNA sequence, UGA, codes for a stop codon, which then prevents the rest of the sequences from being translated, which means multiple amino acids are missing. The formed completely non-functional protein disrupts normal cellular processes, potentially causing genetic disorders, developmental abnormalities, impaired organ function, or increased disease susceptibility. Insertion and deletion mutations are what are known as frameshift mutations. It occurs when the bases are inserted or deleted, and it shifts the entire sequence from that point onward. The original DNA sequence is GCC, which codes for the amino acid alanine. In the deletion mutation, the C has been removed or deleted, and that shifts the codons one base to the left. From that point onward, the amino acids produced are completely different from the original. In the insertion mutation, the extra C has been added or inserted, and this shifts the codons one base to the right. From that point onward, the amino acids produced are once again completely different from the original. Both these mutations produce a completely different protein with different functions that can't fulfill its purpose because the structure has been changed. Mutations are vital as they drive genetic variation and evolution. They create new genetic possibilities for natural selection to shape and enable species to adapt to their environments. Mutations can generate beneficial traits, aid scientific research, and enhance our understanding of genetics and diseases. Without mutations, life would lack diversity, limiting its ability to thrive and adapt. With this, we conclude our exploration into the fascinating world of mutations. Hope you had fun diving into the topic of mutations with me, the one and only Narai. Till next time, bye!